Hi everyone, my name is Rana Ghafouri and I'm completing my master's with Dr. Pia Wintermark in the NeoBrain Lab. Today I'm going to be talking about how sildenafil promotes white matter recovery in the cerebellum following term neonatal hypoxia-ischemia. Neonatal encephalopathy caused by birth asphyxia is one of the leading causes of neurological injury and death in term newborns worldwide. There's a variety of reasons this can happen, and the cause is often unidentified. Surviving infants often experience many neurological deficits, including cerebral palsy, developmental delay, visual impairments, and epilepsy. Currently, there is no established neurorestorative treatment for neonatal encephalopathy and hypoxic ischemic injury. There's been a lot of research done on injury to the cerebrum resulting from hypoxia-ischemia. However, the cerebellum has been understudied in this context. Because of its very unique structure compared to the rest of the brain, it's important to study mechanisms of damage to the cerebellum, which is the focus of my research project. Cerebellar injury has been associated with a number of neurodevelopmental delays related to motor function as well as cognitive disorders. In fact, cerebellar injury has been detected both in term newborns who've undergone birth asphyxia and in rat pups in a model of hypoxic ischemic injury. Our lab has been studying sildenafil as a potential neurorestorative treatment option to repair this brain damage. Sildenafil is a selective inhibitor of phosphodiesterase 5, resulting in elevated levels of cyclic GMP and thus prolonged vasodilation, as well as other downstream effects of the nitric oxide pathway. Findings from our lab suggest that sildenafil promotes neurogenesis and synaptogenesis and decreases neuroinflammation in the cortex in a rat model of term neonatal hypoxia-ischemia. This is the well-established Vannucci model, where the rat pups undergo a ligation of the left carotid artery to induce ischemia, followed by 2 hours in a hypoxia chamber exposed to only 8% oxygen. This is done in Long Evans rat pups at postnatal day 10, which is term equivalent age. Hypoxic ischemic rat pups were randomly assigned to either a 50 mg per kilogram dose of sildenafil or a vehicle, which was given every 12 hours for 7 days, starting at 12 hours post-surgery. We also had a control group of animals who underwent a sham surgery and were given vehicle. The cerebellum was collected at 3 time points: postnatal day 12, day 17, and day 30. Western blot analyses showed that hypoxia-ischemia resulted in decreased expression of myelin basic protein, a protein that is crucial in the process of myelination, at all 3 time points, and sildenafil reverted this expression back to sham levels. Similarly, oligodendrocyte transcription factor 2, a precursor to oligodendrocytes, was decreased following HI and returned to normal levels with sildenafil. Interestingly, both MBP and OLIG2 showed improvement at P12, but had better improvement at the other 2 time points, suggesting a benefit of dosage since the P12 animals only received 2 doses of the drug, whereas the P17 and P30 animals received 14 doses. Finally, there was a similar decrease in expression of SOX10, a marker of mature oligodendrocytes, following HI, which was also reverted to sham levels with sildenafil. In summary, hypoxia-ischemia reduced the expression of myelin and oligodendrocytes, and treatment with sildenafil returned these back to levels that were no longer different from healthy control animals. These findings suggest that sildenafil after hypoxia-ischemia may contribute to recovery of white matter damage, and thus may be an effective candidate for cerebellar injury following hypoxia-ischemia. I'd like to thank my supervisor, Dr. Pia Wintermark, as well as my advisory committee and other lab members. As well, thank you to HBHL and all our other sources of funding.